And of course, it's my pleasure to be back here. So we have 2 talks on dystrophin restoring approaches. And the way that Linda and I have divided it is that I will explain the mechanisms, I will give an overview of what's currently in trials, what the challenges are, and then that will set the stage for Linda to explain the current work that's ongoing to overcome these challenges and to improve all these approaches. So if you were in the earlier session on genetics, you will have heard already about dystrophin, and well, given that you're in this conference, you will have heard about dystrophin anyway. So Duchenne is caused by a lack of dystrophin. And what does dystrophin do? Dystrophin connects the inside of the muscle fibers, the skeleton of the muscle fibers, to the connective tissue layer that surrounds each muscle fiber. And it does that by 2 important clicking domain, and in that way it provides stability to the muscles during exercise. And I like to use the carabiner example to explain what dystrophin does, because the carabiner also has 2 clicker domains, and also you don't need dystrophin to use your muscle. You don't need the carabiner to go rock climbing, but if you make a mistake, you're very happy that you have it. And what is the case in Duchenne is that only the first clicker domain is made, so of course the connection to the connective tissue surrounding the muscle fiber is not there, and therefore, lacking this connection, during the use of muscle during contraction, there's continuous damage, and that leads to chronic inflammation, the formation of scar tissue and adipose tissue. Regeneration is impaired, and that leads to chronic cycles leading to loss of muscle tissue and loss of muscle function. And if you're here as a family, you know the consequences. There is another type of disease caused by mistakes or mutations in the dystrophin gene, and that's Becker muscular dystrophy. And as you can see, the clicker domains are there, only the part in the middle is shorter. Now, Becker is a less severe disease than Duchenne. Duchenne is very severe. So all these processes that I'm discussing, they happen in Becker as well as in Duchenne, only slower. So, since all the problems that go wrong in Duchenne are because there is no dystrophin in the skeletal muscles, an obvious solution would be to restore the dystrophin. And there's different ways in that that can be done. Gene therapy, stop codon read-through, exon skipping, and these 3 are all approved around the world. And then genome editing, which is in preclinical stage. So I'll explain for each of them how they work, what the current state of the art is, but also what the challenges are, because none of these things are cures. First, for everything, there is a challenge, and that is that muscle is very abundant. So 30 to 40% of our body is muscle, and then we don't have one muscle, like we have one liver, we have 2 kidneys, we have more than 700 different muscles. If you want to restore dystrophin, it's not enough that you restore it in one muscle fiber, in one muscle. You want to restore it in the majority of fibers, in the majority of muscles to have an effect. Then also, this process of muscle being replaced by fibrosis and adipose tissues, that is irreversible. So once that has happened, currently we cannot undo that. We cannot bring back the muscle. So that means that the time of intervention matters. And what I'm not saying is you can only do this in very young patients because then you will have the highest therapeutic effect. Whenever patients have function, treating them hopefully can maintain or slow down the decrease of that function. However, it's not realistic to think that if you have a non-ambulant patient and you do gene therapy, that patient then will become ambulant, because the muscle in the leg sadly has been replaced irreversibly. So going over the different therapeutics approaches one by one, starting with the gene therapy. So what is gene therapy? The idea of gene therapy is we don't have a functional code, a protein code for dystrophin. What if we provide the cell with a code that we manufacture, we give the dystrophin code, and then we deliver that to the cells? As said already, it's not enough if you do that to one muscle cell, you need to do that to the majority of muscle cells. So we need a vehicle, we need a delivery tool. And the best tool to deliver genetic material is the virus. And there's actually not that many viruses that can go to muscle, but there is one, and that's called adeno-associated virus, AAV. So AAV is a virus that if you get infected, you will not get sick. So that is nice. However, you will get an immune response. And once you've seen AAV, if you now do gene therapy where we use astronomical doses of AAV, if you've seen it before, there will be a huge immune response. And the immune system doesn't realize this time it's a good virus. This virus has good intention. It just sees the virus and thinks, this is a pathogen, I need to destroy this. So it's important, if you do gene therapy, that the patients haven't seen the AAV before. AAV is small, and this is why it can infect muscle, but this is also why only small genes fit. And again, if you were at the previous lecture on genetics, the dystrophin gene is one of the largest genes in the body. So the gene code doesn't fit. So what we need to do, we need to have sort of a minimalistic approach to make sure that all the components that we need to have in this gene, in the virus, that are there. We need some viral sequences because our gene needs to go into the AAV. You can see the viral shell there. It needs a promoter. That's a volume switch, so the gene is on in muscle and heart. And then we need our dystrophin gene, but that's too big, so we had to make it smaller, and now it's become a microdystrophin. And it's only 32% of the normal dystrophin. So what I just showed, we have Duchenne, we have Becker, where you have a partially functional protein. The Becker proteins are 60% and more of the total dystrophin. This is much smaller. But of course, we hope that it is still functional. So what do these microdystrophins look like? And again, you can see the full-length dystrophin, the two clicker domains. You can see the microdystrophin with this very tiny clicker domain. And then you can see the 4 different microdystrophins that are currently tested in clinical trials. And what you can see, they all have this ABD, actin binding domain, that's the first clicker. And they all have this cis, that's the cysteine-rich region, that's the second clicker. And then you can see that in between this elastic domain, there's some variability. Some have different parts than others. However, you can also see there's more similarities than differences. So if you ask me which one is best, well, if you ask the companies, of course, the companies are going to say that the ones that they have developed is the best. If you ask me, there's more similarities than differences. So if one works, probably the other ones will work similarly. What you can also see is that the different AAV numbers, so there's different AAV serotypes, like the COVID virus, you have the Omicron, you have the Delta. AAV comes also in different serotypes. 7, 4, 8, and 9 all go to skeletal muscle, but they have slightly different protein shells on the outside. So what we know so far, anywhere between 150 and 200 patients have been treated with AAV microdystrophin. And we know from that there's some general things. The first is that if we deliver the AAV microdystrophin, the skeletal muscles of patients will produce microdystrophin. And that is something that 10 years ago we did not think would be feasible. If you deliver it in the bloodstream, skeletal muscles would be able to do this. The levels vary between 5% of the fibers making about 1% of microdystrophin to more than 80% making, well, over 70% of microdystrophin. However, it's clear that this is possible now. So that is the good news, and really something that, well, we didn't think was possible a decade ago, but science has advanced. What we also know is that there are side effects, and these can be severe to very severe. So far, 2 patients have died. They were older patients, but I just want to make sure that you don't underestimate, don't trivialize these side effects. They can be very severe. The side effects are related to the dose. So the higher the dose, the more virus you give, the higher the chance that you get a very severe side effect. So then you say, well, maybe then just give a lower dose. Well, sadly, the lower dose is when you have this 5% of fibers making 1% of microdystrophin, where we think, well, is that enough to have a therapeutic effect? Well, probably not. So you need the higher dose to get enough microdystrophin, but at this higher dose, there's also the risk of these severe side effects. Side effects are also age-related, and again, that makes sense. If you're older, you're usually heavier, and you give the viruses per kilogram, so if you're heavier, you get more virus. And also, if you're older, you have a more mature immune system. The Sarepta microdystrophin has been approved in the US for 4 to 5-year-old patients, but so far this is only based on microdystrophin restoration. And if you've kept up with the news, you will know that they were waiting for their phase 3 clinical trial to see whether there were functional effects. Sadly, that failed its primary endpoint. So there was an outcome measure, the North Star Ambulatory Assessment, and there was no difference between patients who were treated and who were not treated. There were differences between secondary endpoints, the time to rise from the floor, the time to run 10 meters, but not for everything it was significant. So what we don't know is what the FDA will do now, because it would have been much easier if the primary endpoint was met. So there's no clear signal that this slows down disease progression, but there's also no clear signal that it really doesn't do anything. So, well, we don't know what will happen, but just be aware. It's approved in the US. It was a controversial approval, and sadly, the confirmatory study was not that convincing. So back to these side effects. I mentioned already they are severe, and I'll just make them more concrete. Severe nausea to the point that patients have to be admitted in the hospital because they suffer from dehydration. Liver damage, kidney failure, where patient had to be on dialysis temporarily, sepsis, so very severe inflammation in the blood, and sadly two times death. So not all patients have these severe side effects. Fortunately, most patients recovered, but again, don't underestimate them. Then there's a second type of side effects, and that occurs later. So the first one is acute. The second one occurs a couple of weeks after the patients have been treated, and this is muscle breakdown and myocarditis, so inflammation of the heart. And that is because the immune system starts to attack the cells that make the microdystrophin. And it's only seen in a subset of patients, those with the deletion in the beginning of the gene. And why is that? Because I just said Duchenne patients don't make dystrophin, and we know the immune system will respond to proteins that are foreign. So if Duchenne patients don't make dystrophin, and now suddenly cells start to make microdystrophin, you would expect that the immune system would attack all for all the patients. That's not what's happening, and that is because when I say they don't make dystrophin, I mean they don't make dystrophin in the skeletal muscle and the heart. There are other dystrophin forms that are expressed, for instance, in the brain. And these are very small, so they're the last part of the dystrophin, so just this part, which is also part of the microdystrophin. 99+% of patients make this part, so that part is not new. This part of the microdystrophin, so far all patients treated in clinical trial will have seen. Now, most patients have a mutation somewhere here in the middle. So they can make this first part. It's not stable, it's not productive, but the immune system will have been exposed to it. So this part of the microdystrophin is not new for them, except for the roughly 10% that has a deletion in this area, because they cannot make this protein. And now when you provide this microdystrophin, the immune system sees a protein that it's never seen before and starts to attack the cells expressing it, and that's sadly the skeletal muscle and the heart. And of course, that makes things worse, because you're not only losing your transgene, you also lose skeletal muscle and you get an inflammation of the heart, which is not a good thing. And that is why these patients are now excluded from participation in trials and also cannot get the microdystrophin gene therapy. And that's not to discriminate, that is to prevent that they actually get worse because they start breaking down their muscle and their heart. So that is one challenge applying to a subset of patients, but there's also other challenges. And the first one is, how functional are these microdystrophins? We know they're functional in mouse and animal studies. We had a question in the previous session, well, how relevant are these? Well, to some extent they're relevant, but you see this mouse is standing up. That's not usually what a mouse does. Usually it's on 4 legs. And then the dynamics, the forces on this muscle are much different than in a human who walks on 2 legs. So the placebo-controlled trials are ongoing, and so far there's none that has exclusively, robustly shown that there is a slower disease progression. So we need to wait and see. What we do know is it will not be there permanently. The microdystrophin is not a normal dystrophin, it's a much shorter dystrophin. We know from Becker patients there's also pathology, there's turnover of muscle. So with time, the microdystrophin will be lost because there will be turnover of muscle. The microdystrophin doesn't go into the genome, it floats around in the muscle. With time, it will disappear. And then the question is, will it disappear in 2 years, 3 years? Or in 10 years or 20 years, or even more. We don't know that. We do know with time it will go down, but we don't know when. Then you say, well, can we then retreat the patients after it's gone down to 0 or to too little? No, we cannot, because by the time you've treated the patient, there will definitely be an immune response if you come back with AAV. So that is also a challenge, and I went over the immune response to microdystrophin. So there's work to be done still for this, but let's move to the second one, and that is stop codon read-through. So stop codon read-through is for a very specific type of mutation, and that's the so-called stop mutation or nonsense mutation. So we have here the genetic code of the dystrophin protein. We have a start to tell the cell, this is where you start the translation, and at the end we have a stop. Protein is ready, it can be released into the wild to do its thing. Now, a stop mutation is a mutation where one piece of code that normally would code for an amino acid is changed and now becomes a stop code. So this code doesn't make sense because it's in the middle. I mean, why would you make this whole transcript, this whole code, when the stop is in the middle? Well, what's this doing there? But the cell still uses this stop. So what you now get is only the beginning of the protein, which is not functional. There are compounds that can suppress these stops. So not this one, the original one, that's normal, just these ones. And that is the basis of stop codon read-through. So that only works for nonsense mutations or stop mutations. You can see here, this is a stop mutation. You see the genetic code is not disrupted. There's just this one change here that causes a stop, an asterisk here. Every mutation that has deletion or duplication in it, whether it's small within an exon or whether it's large, a deletion of exon X, Y, or Z, duplication of X, Y, or Z, these are not eligible for stop codon read-through. So about 10 to 15% of patients do have these nonsense mutations, however, and for these there is stop codon read-through, and the compound is called ataluren or Translarna, and it was approved by the EMA in 2014, conditional approval, and currently it is approved conditionally for patients 2 years and older. In the EU, it's marketed in European countries, the UK, several other countries as well. But because it's conditional approval, there's evaluation every year. It's only for nonsense mutations, I said that already. And then there's 2 confirmatory studies done, and sadly, these did not confirm a slower disease progression, only in subgroups. And regulators don't really like that type of analysis. There is also a registry. So since this was approved in 2014, as a field, we could start collecting the data of patients who were on this drug commercially. So there's years of data collected. And if you look at the STRIDE registry that collects this data, there is a delay of disease milestones. This is not something you can test in a clinical trial. Clinical trial is usually a year to 1.5 years. And then, well, if you look at 100 patients, often there's maybe 1 or 2 that lose ambulation. You can't really draw conclusions. Now we have hundreds of patients and we look for 5 years or more. And what is seen is that compared to the natural history, because you have no placebo group in this registry, compared to natural history, there's a 3-year delay in loss of ambulation, and there's also a slower decrease in the forced vital capacity, so the lung function. The CHMP, so the Committee for Human Medicinal Product of the European Medicine Agency, however, voted against renewing the conditional approval because they said, well, this confirmatory study didn't show a slower disease progression, and well, they said they took the STRIDE registry into account, but they didn't find it convincing. PTC will appeal against this. I think for the UK, for now, you're no longer part of the EMA, so I don't think for you that this matters currently. What's happening in Europe is that as long as this appeal is pending, patients who are treated remain on treatment, but of course this is not a happy development for the field. So that about the ataluren. We're going to move to exon skipping. And exon skipping tries to make the code readable. So for these mutations where ataluren doesn't work because the code is disrupted, exon skipping might offer a solution. So what you see here is a genetic code. So the genetic code is divided over exons, and these all have shapes. And what you can see here, exon 48, 49, and 50 are missing. So 47 now is linked to 51, and you see this doesn't fit. You see this does fit to 52. So there's a process in the cell where all these exons are linked together, and what we actually want with exon skipping, we want the cell to ignore this one, because then 47 would be linked to 52. And we know that when 47 is linked to 52, this gives rise to a partially functional dystrophin, because we've seen these mutations in Becker patients. Now, how do we get the cell to skip this? We use antisense oligonucleotides, AONs or ASOs, that specifically bind to a target exon. So they're small modified pieces of DNA that specifically bind to an exon, hide it from the splicing machinery that connects all these exons, and now 47 is linked to 52. And the code is restored, as you can see, and then you produce a Becker-type dystrophin instead of a non-functional dystrophin. So where are we at with exon skipping? There's 4 approved drugs in the US, 1 in Japan, and all of that. So everything that's approved in either the US or in Europe is also approved in Israel. So there's 4 AONs, but they target 3 exon because there's 2 approved for exon 53. This applies to about 30% of patients. So this is, again, a mutation-specific approach. Depending on what deletion you have and where you have your deletion, you're either eligible, yes or no, and you need a specific exon to be skipped. Exon skipping approval is based only on dystrophin restoration at low levels, so less than 1% to 5%. What we don't know yet is whether this is enough to slow down disease progression. And you will probably hear a pattern here that reminds you of what was true for the gene therapy approach. The first exon skipping drug was approved in 2016, so that's 7 years ago, and so far there is no evidence that there is a slower disease progression. So we need to wait for these trials. They are ongoing. What is clear is that there's room for improvement, because, 5%, if it slows down disease progression, 10 or 20% is going to be better. So we need to improve. What do we need to improve? The delivery of these compounds to muscle and heart, because that's probably why we get these low restoration levels, but also the applicability, because we do this for different exons and they all apply to small groups of patients. So how can we as a field improve uptake? We can say, well, just make sure that the AONs get better to whatever tissue, then they will also go better into skeletal muscle and heart. You can do this by linking arginine-rich peptides to them. And there's 3 companies developing this, Sarepta, PepGen, and Entrada. The question is, how tolerable are we as humans to these arginine-rich peptides? Because we know they can lead to kidney toxicity, and then the question is, will they restore dystrophin at a good level before they start leading to kidney toxicity? So that is something that the clinical trials will have to reveal. Then there's also companies that say, well, instead of uptake in general, we want them to specifically go to muscle. So we'll either deliver an antisense gene with AAV, or we add a transferrin receptor antibody. And transferrin receptors are expressed on muscle. So if we add this antibody, then the AONs will specifically go to muscle. And Avidity and Dyne are looking into this. Challenge with this is antibodies to antibodies. How specific is it? Transferrin is expressed on muscle, but also on brain, also on other tissues. So that's we'll have to wait and see. And for this one, well, this is AAV. So all the side effects that I mentioned earlier apply here as well. So if we look at what's going on in the clinical trial field, what you will see is that there is a lot of focus on the first 3 exons. So we have 3 exons for which exon skipping is approved. There's confirmatory trials ongoing. There's new trials ongoing to try these arginine-rich peptides, these transferrin receptors. There's trials planned. And then what you can also see is that, well, there's some companies focusing on 44 for which nothing is approved yet, but then there's a lag. And you can see the percentages go down. So it becomes really difficult to do a clinical trial if only 3% of patients are eligible for a certain skip. However, if you have six companies all trying to go for exon 51 skipping, that's 14%, you can also see that the percentages there become really small. So this is currently a challenge where, as an academic, ideally I say, if the companies would just have a gentleman's agreement, you go for this one, I go for that one, then they wouldn't have to fight. But of course, well, I can live in that utopia, but that's not what's happening exactly. So now comparing exon skipping with microdystrophin, I'm not going to say one is better than the other. They are different, and I just want to show you how they are different. They're both approved, but they're both approved based on dystrophin restoration. There's no functional evidence yet that either of these approaches slows down disease progression. For exon skipping, we know that the dystrophins that we produce are functional because we see them in Becker. However, we produce very low amounts. For microdystrophin, we don't know yet whether it's effective in humans, but the amounts that we produce is much higher. Exon skipping is mutation specific. The microdystrophin is not. There's about 10% that are excluded because they have a mutation at the beginning. Most patients, however, are eligible. Microdystrophin AAV, you need to give only once, or you can give only once. So side effect-wise, yes, they are more severe, but only once. While the regimen currently for exon skipping is weekly intravenous infusions. The duration of effect, well, for the exon skipping, as long as you keep giving it, there will be an effect. And we know for the microdystrophin, at some point, the microdystrophin will go away. What we don't know how long this will take. And I mentioned the side effects already. Yeah, I will wrap up. Genome editing, very briefly. I'm sure it's hyped here as well. What is genome editing? Genome editing is a scissor that cuts in the DNA and a GPS that tells it where to cut. Now, why would you want to cut the DNA? You want to cut the DNA because you activate the repair mechanism. There's one that can repair things perfectly, but we're not going to use that because it doesn't work in skeletal muscle. So we're stuck with the glue system that just sticks things together and creates errors. Now, why would we want to create errors? Because then on DNA level, we can make an exon invisible. And if we make an exon invisible, then rather than having to do the weekly injections with the AONs, every time the DNA produces a transcript, this exon is not included. So it's exon skipping on DNA level, so to say. So it's an early stage. I know everyone is hyped because it got the Nobel Prize, but it got the Nobel Prize for chemistry, not medicine. There's therapeutic potential for Duchenne, but the effect depends on the time of intervention. Again, it cannot undo damage that's already been done. You need to deliver it, and it will produce a partially functional dystrophin. That goes for everything that I mentioned, but all the other approaches, no one says CRISPR cure. Well, some people working on CRISPR, they claim that the CRISPR is going to cure. It won't, sadly. So there's things that we need to improve, we need to deliver, we need to make sure it's safe, that it doesn't edit where we don't want it to edit. And we still have the immune system to AAV, but also to the protein involved, so the Cas protein, the scissor protein. I'm going to skip this and just leave this summary up. If people want to take a picture, I'm not going over it in view of time, but I'm going to leave you with this and then hand over to the next speaker to give her talk. Thank you.
Her slide. Thank you.
Okay. Hello, everybody. Hopefully my slides will come up. Oh, yeah, they're there. So, yeah, I'd like to thank the organisers for inviting me and to Annemieke for setting the scene so wonderfully. I've got a big job to try and address all the challenges, so I won't actually address all of them because I'll be here until 5 o'clock and you'll get a bit bored, I think. So I'm going to do some snapshots if that's okay. Oh, I'm sorry about that. I don't know why you can't see the titles. Anyway, so this summarises hopefully the things that Annemiek's just gone through. So on the left there we have the exon skipping, and the issues, as she's gone over, is the applicability, so it's a mutation-specific therapy. The efficacy is quite contentious. We're getting very low levels of dystrophin restored. We obviously need to enhance the delivery, and there's a need for repeat administration. The stop codon read-through, that's the second one along, it's the applicability again, mutation-specific, and the efficacy. So Annemiek's gone through how the approvals have not gone through from the conditional side of things. For the AAV, with the microdystrophin, which is the third one along, we have the immune response, we have toxicity because we're putting in a high level of virus into patients. The protein isn't as functional as it needs to be, and there's a really high cost with this therapy. So I think it's something like $3 million per patient, so we need to improve the cost side of things for patients. And then this is the gene editing on the right, so this is the cutting of the DNA. Again, it's mutation specific. There are safety, efficacy, and delivery issues. But like I said, I'm not going to go through all of those. Annemieke has covered some of them, so I'm not going to duplicate. But I will focus on just 3 of them, so exon skipping, the gene addition, and the gene editing. I won't touch stop codon read-through, if that's okay. So for the exon skipping, so just to go through again, so these are the issues. And for me, these are the things that I think could be potential solutions. So for applicability, obviously we could maybe use a different therapy. So the microdystrophin might be the other choice. But we obviously need to consider those rarer exons where there's only a handful of patients who might be treatable. So we should be optimizing and putting through trial the antisenses for those exons. And I know there's some foundations in the US that will do that, so almost for n equals 1 patients. And for that coming to the UK would be amazing. For the efficacy, Annemieke went through that, so I won't repeat it, but we need to improve the delivery and reduce the clearance. That's using the antibodies and the cell-penetrating peptides. And they will also improve the delivery concerns, so they should hang around for longer in the muscle. So we might be able to go in bi-weekly or monthly instead of having to do a weekly dosing. We can also use the AAVs, which may give longer-term expression, so it might be a few years that they stick around for. So these are all, could be used as strategies to improve the delivery and the administration. So what I will focus on is just alternative chemistry, because I think Annemieke covered most of the rest of it. So I'm sorry about the slides. I don't know why they've all shifted the format. Anyway, so to improve efficacy, we can look at changing the chemistry. So the approved antisenses at the moment are all based on a particular chemistry called PMOs, but we can change that and we can maybe enhance the delivery and also the efficacy. So stereo-pure antisenses, so these have been developed by Wave, who are sponsoring the conference. So here they're looking at the bonds in between the bases that make up the antisenses. So they get them to point in certain directions. So you've got some pointing up, some pointing down. And they've analysed this and they show that they can improve the efficacy by using these stereo-pure antisenses. And very recently, in October, at the World Muscle Society, they presented on their exon 53 trial. So they went through animal models and they showed the animals that they get enhanced exon skipping and it improves survival and rescues respiratory function. They also showed in non-human primates that they get high levels of delivery into the heart and diaphragm. So that's all very positive. And in the trials, I don't think the pointer works, but I'll try. In the trials, they show that they get 53% exon skipping after 3 doses of their antisense. So it's looking good. It looks like it sticks around in the muscle so that we can go in on a 2-weekly basis. And they're continuing into the phase 2 trials, which are expected to start soon. And there might be results in next year. So this is all very positive. So there's also tricyclo-DNA antisenses. So tcDNAs have been around for a while, but recently, in the last few years, the group of Aurélie Goenval have applied these to Duchenne, and they've tested them in the MDX mouse model. So this is what the chemistry looks like. It looks quite complicated, but it's just got these 2 rings, which is the important difference to what's used at the moment in patients. And they've done lots of work, and they show that the antisense crosses the blood-brain barrier and restores dystrophin expression in the brain, and that it has a positive effect in the mice that are treated, so ameliorating the emotional deficits. So again, this is a really important step forward for the use of antisenses for Duchenne. And the company Synthena are planning a clinical trial. So recruitment has started, and this was in March this year. So we'll see where that goes once they release some results and start the trial. And then finally, there's mixed backbone. So not just having one chemistry in the antisense, you could have a mixture. And again, this is quite dated work, but they have done mixtures of chemistries, and they show that they can increase the levels of dystrophin-positive fibers in the MDX mouse. So, there is potential to change how well the antisense works by just mixing the backbone. Obviously, there's a lot of permutations because the antisenses are 20 to 30 bases long, so there's a lot of differences you could examine. But WAVE has also worked with the Oxford lab to test both the combination of the mixed backbone and the stereo pure antisenses. And I mean, it was a very complicated paper, and there's lots of graphs and figures, but basically they're showing with various different bases at different points and the different stereo pure elements, they can again increase the levels of exon skipping. So we'll see WAVE, obviously they're involved in this, so they may take this forward into clinic as well. So BioMarin, they were involved in a trial for exon 51 skipping, and they now have a new antisense for exon 51, where they have, we think, a mixture of chemistries, or it's modified slightly. I'm not exactly sure what the difference is, but this is again going into clinical trial. So there are big steps happening in trying to improve the antisense itself and get better exon skipping. So I'll now move on to gene addition. So these are the problems summarised, and Annemieke went through them very nicely. So there's the immunity. Obviously, if a patient has been exposed to the virus before, they will have an incredible immune reaction, so they're now excluded from trial. But you can have an immune reaction against the microdystrophin. So there are potential steps you could take to tolerize the patient by, exposing them until they don't recognize it as foreign any longer. You can use immune suppression. The ideal would be able to go in with a different serotype, and I'll go through that in a moment. But ideally, would go in with a lower dose, and an earlier treatment, so that we're not having to expose the patient to such high doses of the virus. The toxicity is related to the dose, so again, going in early is better. And like I say, the new serotypes, which I'll take you through. And ideally, we would have expression of the microdystrophin in the muscle and not anywhere else, so we can use synthetic promoters. And there's companies looking at developing those. The protein isn't as functional because it's a microdystrophin, and I'll go through how we can improve that. So these are things I'm going to concentrate on. So looking at the serotypes, so as Annemieke pointed out, the serotype is determined by these proteins that are expressed on the surface of the antisense, so they stick out, and that's what's recognized. So we can change those by creating these AAV libraries. So basically you take what you know as good serotypes for targeting muscle, and you can shuffle them up and make these libraries and then test them to see how they deliver and transduce muscle. You can also make specific mutations and insertions and change the chemistry. So again, they don't get recognized by the body. So we can do all of this, and many companies are working to develop these novel serotypes. And I think that will be what comes through next in the gene addition therapies, not just for Duchenne, but for other diseases as well. So now moving on to the functionality. So as Annemieke pointed out, it's a very small protein that we're trying to get expressed. And if you compare what the full-length dystrophin looks like, so it's in the grey here with the different regions. So this is the N-terminus that binds the actin, but you'll see that actin also binds two of the rods in the rod domain in the middle, and in the microdystrophin that's missing, so we've only got one connection to the actin. We've also lost the extra connection with the microtubule, which is important in the contraction, so we've only got one connection. And we've also lost in some of the microdystrophins this connection with the N-NOS, so this is neuronal nitric oxide synthase, which is important for dilating muscles during contraction. So this isn't in the microdystrophin, in the one that's approved. And the important other loss is the connection with all these other proteins at the muscle membrane. So these are all connected here, but we've lost that. These are all non-connected to the dystrophin. So it's a really severely compromised protein that we're expressing. So we can try and address that by delivering more. So there's been lots of work using what's called dual and triple AAVs. So here, a Becker-like protein that's, or coding sequence that's longer than the microdystrophin is split into two, and each half is then packaged into its own AAV, and then you deliver both together, and the signals between will join them together, so you get this longer protein than what is expressed with the microdystrophin expressed in the muscle, so it's more functional. This is work that was done in the lab I was in, so we can also divide the full-length dystrophin into three and package again each one into its own AAV, and then these will get spliced together, joined together to make the full-length dystrophin. And we show in MDX mice for both of these strategies that you do get expression, but it's obvious that this isn't going to go near patients because a single AAV is causing some severe toxic side effects. Going in with three, we'll have to lower the dose, so the likelihood of it being effective is really small. So this is probably unlikely to go any further. So there needs to be other methods, so look at other delivery vectors. So exosomes are really nice, naturally occurring membrane-bound vesicles that are basically released from all your cells in your body, and they act as sort of messengers between cells, carrying message. So we can harvest these and then pack in our cDNA, the coding sequence of the dystrophin, the full length, because they can hold that size, and then deliver them back into patients, and these will target the cells, and we can decorate them as well with specific proteins so that they will go to muscle only. So these are being explored by Evox. I believe they're still working in this space. We can also make our own sort of exosomes synthetically. So these are called nanoparticles. And basically you mix together lipids and proteins and they form this sort of shell. And if we give it the cDNA of the dystrophin, the full-length dystrophin, we will form this shell. And again, we can decorate it with proteins that will tell it to go to muscle. And 4Base Bio were working on this with us, and hopefully it will continue. So non-viral vectors are probably going to be really important in the future. We can also use other viruses that have a higher packaging capacity. So this is, I'm sorry I haven't got all my titles on here. So this is herpes simplex virus. So that causes cold sores. And generally, it seems to be quite toxic using that as a gene delivery vehicle. It's now approved for a skin condition, but we can, but Replay have developed a method for making it to be a safer virus, so that they are envisaging that it could be given systemically, and that would hold the full-length dystrophin. We can also use these so-called bacteriophages. So a paper came out very recently. So bacteriophages are what are produced by viruses to eat up bacteria, and we can modify them to target muscle by changing the proteins that are expressed on the outside. And again, because it's got a high packaging capacity, we can put the full-length dystrophin in. Like I say, there was a paper recently on that, and I know Replay are very interested in trying to exploit that further. So now moving on to gene editing. So as we've already gone through, so these are the problems. So at the moment it's being developed as a mutation-specific therapy, but we can try and enhance this by targeting around hotspots, integrating super exons, and integrating at the start of the dystrophin gene to make more universal. I will go through that in more detail. We obviously need to improve the efficacy by improving the delivery, and as Annemieke said, we can use AAV to deliver, but that obviously comes with its own problems, but we can also use the non-viral vectors, so the nanoparticles. For the safety, I won't go through all of this because it's quite complicated, but for the safety, the main issue is that it's not as specific as it could be, gene editing. It has very high off-target effects, which can obviously be very detrimental because you're cutting DNA, and the repair that happens could end up being knocking out a gene or switching on a cancer gene, so we need to improve that. So we can use, so the commonly used Cas is called Cas9, SP-Cas9. but we can use different ones which are more stringent, means they don't have as many off-targets. We can also just cut one strand of the DNA rather than both, so they're called nicases. And we can also delay the expression, not delay, but cut down the length of time that the Cas is expressed, so we can deliver protein instead of the gene for the Cas. And we can also get the Cas just to be active in the muscle by using muscle-specific promoters if we're using the gene. And as Annemieke said, there is an immune reaction to the Cas itself because it's from bacteria. So patients will have seen a bacterial infection, potentially, that carries the Cas that we plan to use, so we can immune suppress. So just to go through the applicability side for the gene editing. So, as I said, we can target around mutation hotspots, integrate superexons, and then do mutation agnostic strategies. So this is the mutation hotspot. So there's a hotspot between exons 45 and 55. So if we use Cas to cut before exon 45 and then after exon 45, in theory, this end that's cut here could be joined together with the end that's cut here, and we get that big deletion of exons 45 to 55 removed, and then we'll have a working gene. And this therapy would be applicable to 62% of patients. But it's not going to be completely effective because when you think about it, this cut here could easily be repaired there, and these two ends joined together, and we could have these two ends joined together. So it's not as efficient as we would need, but it has worked in cells. So it'd be interesting to see how this moves forward. For the superexons, so this is work of Charles Gabash. He did the hotspot mutation work as well. So here we're relying on a repair mechanism that should be active in muscle. So it's a process called HITI, which just basically means we're supplying this repair template that carries exons from the mutation, so it's deletion of exon 52, and we're carrying exon 52 to 79, so it's the full length of the gene from the mutation onwards. And we can use the Cas to cut either side of this coding sequence that we want to integrate. And because of that cut, we will then have these ends that will be seen in the DNA that we've cut in the dystrophin, and that will then lead to this being integrated. I'm sorry if I haven't explained that very well, but basically we're asking the cell to insert this in the cut site by providing the right sort of template. And Charles was able to show that he can actually get dystrophins. This is the protein. On these two here, it's the superexon insertion. We are getting protein expressed, but again, it was really, really low efficiency. So there's much work that needs to be done to improve that. And then moving on to the mutation agnostic strategy. So this would mean that we could try and develop a gene edit that could be applicable to as many patients as possible. So there's two strategies that we've worked on in the lab. The first is to try and integrate into what's called a safe harbor site. So these are sites within the chromosomes that will allow you to put in a DNA, but it won't be detrimental. It won't switch on genes around it, so it's much, much safer. So we have looked at that, and we can show that we can integrate into that site and we get protein expressed. We've also looking to target the dystrophin gene itself, which would be the ideal, because we then have the dystrophin promoter drive an expression of what we're trying to insert. And I really apologise for my slides, they look awful. But basically here we're targeting at the start of the dystrophin gene with our scissors, our molecular scissors, and then when we provide a repair template to knock in it was a microdystrophin that we were trying to knock in, but we could adapt it for the full-length dystrophin gene into this start of the exon. And we're able to show at the RNA level, which comes before the protein, that we get the integration, and then here's the protein, very faintly, but we were getting microdystrophin protein expressed as well. So it would be interesting to see if we could develop this further. And then, I'm sorry, but everything's missing off here, but I'll say the words that are supposed to be here. So the things that we need to consider for all of these is the timing of treatment. So as Annemieke has pointed out, We need to go in early because the muscle is the most preserved at that point and will have the highest benefit. But we also need to consider what we do for patients who are older, where the muscle is fibrotic. We can also consider the use of combined gene therapies. So, maybe combining an exon skip with a gene addition therapy. So there have been studies in the MDX mouse where that has been explored, and it shows that they work very, very well together, and there's no reason why that couldn't happen in patients, obviously with the right mutations. And as we heard from Esther before, targeting the inflammation and the fibrosis along with the gene therapy could also be extremely beneficial, and that will probably be what comes through for the maximum benefit. And then the ideal is for these gene therapies, particularly based on the AAVs, is to target the satellite cells of the muscle. So they're the cells that will divide and then fuse and replace the damaged muscle. So because at the moment the AAVs that are used don't target the satellite cells, the replaced tissue then won't have the dystrophin in them. So we will end up losing the dystrophin that way as well. So we need to be targeting the satellite cells. So that will come through from developing the new capsids and exploring if we can get something that targets the satellite cells much better. And with that, I will finish, and I apologize for my slides.
Thank you. Thank you very much.